A real exciting fellow seminar planned for this, well, I guess it's kind of afternoon now. And then followed by a symposium on allosteric modulators later this afternoon for some of you who are staying. So it's my great pleasure to introduce Dr. Michelle Glass, who heralds from New Zealand, the University of Auckland, where she obtained her PhD and rose through the ranks and is now a professor. Her area of specialty is molecular signaling, particularly focused on the cannabinoids and on both synthetic and phytocannabinoids from the plant. And she's going to talk to us today about some allosteric modulators. And without further ado, Michelle.
Thank you. Am I live here? Yeah? Yeah. So thank you very much, Jenny, and I want to thank Tim and the rest of the organising committee for inviting me. And it's an absolute pleasure to be here. I've had a great couple of days, and I'm really looking forward to the rest of the meeting. There looks like there's going to be some great talks, and I'm looking forward to actually getting mine done so we can get on with the new interesting stuff that I'd like to see. So what I want to talk about today is some work we've done with one of the most well-characterized allosteric modulators of CB1. I'm going to start presenting some of the data that is already published and then talk a bit about some of the stuff we have that we haven't published yet. Now, I know that this is a sort of hybrid seminar where some of you are here for the allosteric symposium and know all about allosteric modulators and probably generated half of the data, and some of you are fellows and maybe social scientists. So a brief introduction to what an allosteric modulator is or is supposed to be. So when we're thinking about receptor signaling, this is a schematic of the receptor here, and we have an orthosteric binding site. And the orthosteric binding site is the site that the endogenous ligands bind to. And so most of our classical drugs, our agonists and antagonists, are binding to the orthosteric site and competing with the endogenous ligand for that site. But there are other binding sites on receptors, and these are classified as allosteric, and they're generally thought to be sites which are conformationally linked in some way to the orthosteric site. So they change the response that you see to the orthosteric ligand. And they might do this by changing the affinity of the orthosteric binding, or they might do it by changing the efficacy in response, or they might do a combination of both. And sometimes allosteric ligands also have activity on their own. So that would be allosteric antagonism, for example, or allosteric inverse agonism, where you might have an effect even when the orthosteric ligand isn't present. And so allosteric modulators are a hot topic in molecular pharmacology right now. They're sort of one of the great hopes for drug therapy. And the idea of why they might be more useful is that they might be more subtle, really, than the orthosteric ligands. So if you think about this, with cannabinoid receptors, you have lots and lots of cannabinoid receptors right across your brain. And at any given time, not all of them are going to be active. You're going to have a little bit of endogenous agonist in some brain regions right now, and hopefully not in others. So some of you are hungry, so maybe your endocannabinoids are up, but hopefully in the hippocampus they're really low. So you're not blocking your short-term memory formation. You're going to remember what I'm talking about here. So we want, a variable level. If we put in an orthosteric agonist, we drive every one of those receptors maximally simultaneously. Right? It's not really a natural situation. So yes, you get the therapeutic effect you want, but you're also getting activation of other receptors. The idea of an allosteric modulator is if it's not got any activity on its own, then it's only active when the endogenous ligand is there. So you're selectively tuning up or tuning down the response to the endogenous ligand. So therapeutically, that should be a lot more subtle, and the hope is you're going to get less adverse responses to that. So with CB1, there's now quite a number of different allosterics reported, but a lot of them are starting from this point. This is ORG 27569. It was the first allo reported, and it was reported back in 2005. Subsequently, PSN-CBAM, which has some structural relationship with org, was also reported. And there's been lots of different analogues of these compounds subsequently. And what we knew from the initial studies was that being a cannabinoid receptor, they're different to everybody else, always awkward. And so what the org did was it enhanced the binding of the orthosteric ligand, so you see more binding at low concentrations, but it decreased the efficacy. So it's a bit paradoxical. You get an increase in binding and a decrease in efficacy. And there were some question marks in the early papers about what it was doing on its own, and I'm going to talk a bit more about that as we go through the talk. And so we just set out really to sort of characterize some of this. One of the other things that we always hear about allos is there'll be less tolerance, there'll be less desensitization, and our kind of thinking was, well, will there? If you get more efficacy, surely you get more tolerance. Maybe, maybe not. So we wanted to sort of think about what these did in terms of receptor regulation as well. So CB1, our favorite receptor, very widespread through the brain, has at least 2 endogenous agonists, anandamide and 2-arachidonyl glycerol, and it's G protein linked, and it's linked predominantly to GI. So the activation of it leads to the inhibition of adenylate cyclase and decreased cyclic AMP. It also stimulates inwardly rectifying potassium channels, and following activation it is internalized and then we believe predominantly degraded. After we don't see recycling of CB1 within our model systems at least. And so these are the sort of pathways that I'm going to talk to you about: a decrease in cyclic AMP, increase in potassium, and this internalization pathway. And so because we were looking at cyclic AMP, and I had actually just done a sabbatical in Arthur Christopoulos' lab where they were using this cool cyclic AMP biosensor, so I sort of stole it and brought it home. And what it is, it's EPAC, which is a cyclic AMP binding protein. It's the cyclic AMP binding region of EPAC, and it's got a couple of Brett tags on it. And so when there is no cyclic AMP present and you put in the substrate for the luciferase, you get energy transfer between the two and emission at the YFP wavelength at 535. When cyclic AMP binds, it just pushes these tags further apart, and you get less Brett occurring. And so this has some advantages over your classical cyclic AMP accumulation assays. You don't have to have a phosphodiesterase inhibitor present. In most cyclic AMP assays, you try and prevent the breakdown of cyclic AMP to get a bigger signal-to-noise ratio. So this allows you to do it in real time. You can get kinetic data, and you can get that in a 96-well plate format, so you can get kinetic data with your classical assays too, but you have to do each time point separately. You know, this one, you just watch it as it happens in real-time signaling. And it appears to be pretty sensitive as well. So we've done most of our work with this now. And this is the kind of thing that we see when we're looking at classical orthosteric ligands. So because we're looking for a decrease in cyclic AMP, we have to stimulate the cells with forskolin in order to stimulate cyclic AMP, and then we can reduce that down with our agonists. And so here you can see when we stimulate with forskolin in the red, this is across time. There's an increase in cyclic AMP. It's an inverse Brett ratio such that when it goes up, that is an increase in cyclic AMP. So we add our forskolin, you see this rapid increase, and then it sort of plateaus. CP55940 is a classical orthosteric agonist for this receptor, and you can see that it's inhibiting that response nicely. And so there's lots of different ways you can analyze this kind of data. You can take a slice through, you can do an area under the curve analysis of it. If we do area under the curve, we get data that's very similar to what's been reported for accumulation assays. So you can see here with different concentrations, we see concentration-dependent inhibition of cyclic AMP. ending up with a potency around the low nanomolar, which is consistent with what's been reported by others for these ligands in the more conventional style assays. So then we came to look at this with the Organon compound that I'm just going to call Org, so I don't have to get the numbers wrong every time or different every time I say them. So this is the same style assay as what I just showed you. You've got the forskolin response being inhibited by CP here. And when we put the Org compound in, and this is at 1 micromolar in this picture, what you can see is it seemed to track along with CP for about 5 minutes. And then we saw the receptor, or the signal at least, turning off, and maybe a little bit of inverse agonism as well here. So we were turning the receptor off, but it was going to levels beyond that produced with forskolin. So actually an increase in cyclic AMP beyond this level. And so we thought, well, that's kind of weird, but is it, we're adding the drugs together at time 0 here. Does the Org just take longer maybe to get into the receptor? Does it need to bind first before the CP does? So we played around with this a bit to try and understand this. So we did our classical assay here, and then this is with 20-minute pre-incubation with the Org. You could see it's exactly the same. We still track along for about 5 minutes with CP, and then off it goes. We tried switching it round. We thought maybe the CP needs to get in there first, and then the Org comes, and then it does its thing. But no, so here we've added the CP at time 0. You can see the inhibition and the curves tracking together. Then we add the Org at the 5-minute time point. 5 minutes later, again, off it goes. So it didn't seem to be that. And then we thought, well, maybe, we're right in the kinetic part of the forskolin response. Maybe it's something to do with forskolin coming in and turning on the receptor. And maybe we need to get away from this part of the forskolin curve. So we waited until the forskolin response had flattened out. Then we add both drugs together. You can see that with CP, you get it inhibiting the cyclic AMP, and it stays inhibited. With the Org compound, it tracks perfectly with the CP curve, and then after about another 5 minutes, whoop, it turns it off again. So no matter what we do, we see this 5-minute delay at this concentration of Org. When we looked at different concentrations, and I know this sort of starts to turn into sort of some sort of weird modern art picture at this point, but you can see at here in the purple, pinky color is the 1 micromolar that I've been showing you previously, and then if we go for lower concentrations, that delay is even longer. We're looking out to about 10 minutes, and then it's turning the receptor off. But if we come to higher concentrations, it seems to happen a bit more immediately. So we can see, it sort of, really we discovered this because we just happened to start working at 1 micromolar, I think, and so continued to do the kinetic responses to this. So we do see the delay seems to be concentration dependent, but you can see there that it's still completely turning off the signal even when it's taking about 10 minutes to do it. So then it struck us that the antagonists came along long before we started doing kinetic assays, and so we didn't actually know whether they produced immediate responses or not. So we thought, well, we better just make sure that this isn't just what antagonism of the receptor looks like. So with CB1, we don't have a neutral antagonist. We believe we may have solved that problem on this trip, but we don't have a neutral antagonist. The SR compound is actually an inverse agonist. And so red is forskolin, blue here is the CP response, and then as we add increasing concentrations of this antagonist inverse agonist, you can see even at these low concentrations at every time point you're immediately seeing antagonism. There is absolutely no delay in this signal, and then we go on up above the forskolin curve, and that is the inverse agonism. And that's thought that the receptor is constitutively active, and you can turn it off with an inverse agonist. And because its natural activity is to inhibit cyclic AMP, that translates into an increase in cyclic AMP in the case of the inverse agonism. So clearly what we're seeing with Org is quite different to what these classical antagonists would do. When we submitted this, one of the referees said, of course, you're working in a hexal line. It's all an artifact of overexpression. Go and do it in a naturally expressing cell, which we're glad often they're right. And we've done this, and this is in neuro2A cells, which have the mouse CB1 receptor in them. And you can see again, there's the forskolin response. Here's with CP, and there's the org. And again, we get it tracking perfectly with CP for about 5 minutes and then turning the receptor off. What you'll notice is there is no inverse agonism in this. And I've got SR in there as well. And you can see that in these cells, we don't see any inverse agonism. And we think that that is because there's just really low receptor number in these cells. And I think in order to really detect inverse agonism, you need to have higher expression of the receptors. So that was part of the evidence, really, for the fact that what org is doing is driving an off conformation. It is inverse agonism. We had other studies that I'm not going to show you that were also consistent with that. But we do believe that the stimulation of cyclic AMP that we see with the org is inverse agonism rather than it driving a different signal like GS or something, or some alternate pathway. So I showed you at the beginning that with our classical assays where the curves are all running parallel to each other, we can just do an area under the curve and the data all kind of makes sense. But when the curve is doing something funky like this, area under the curve analysis becomes quite meaningless. So we had to try and come up with a way to analyze this data that gave us the parameters that we were interested in rather than just smushing it all together. And so Tash Grimsey in my lab developed this analysis approach, which was to normalize the data to the CP response. So the CP response becomes a flat line, and then we can see the other responses relative to that. Because what we figured we were interested in is this delay, and how long is it, and is it significant, and that sort of thing, and the plateau up here. Is this actually inverse agonism? Is it an equivalent amount of inverse agonism to what SR produces? And so we came up with this analysis that allowed us to see the delay as the X0 value, as a delay before you start to see the receptor turn off, and then it also gave us the plateau. So it's just a delay followed by an association curve. You see forskolin is sort of immediate, but with the org we see delay and then above the forskolin response. And so when we look at this in terms of concentration dependence, and this graph always completely freaks out your classical pharmacologists because it has time on the y-axis, which is an absolute no-no in pharmacology, but it's time versus concentration. They both always go on the x-axis. One of them had to go on the y-axis, but the pharmacokineticists like hold their head in horror when they see this. But so what we've done is look at what is that delay relative to the concentration of the compound. And we've only analysed it for concentrations of the antagonist or the allosteric modulator that actually do antagonise, because otherwise, obviously, the delay would be infinity. So what you can see here with the org compound, as we saw before, you see a much longer delay at lower concentrations. The PSN was quite interesting because while it's quite structurally similar, it does do the same thing, but the delay is notably shorter, whereas with the SR compound, you see absolutely no delay. And when we came to look at the upper plateau, how far it went above forskolin, you can see that both org and PSN produced very similar responses there, but the SR seems to go higher. Now, SR is high potency, but of course this is in the presence of an agonist, so you're out competing. So we couldn't actually get to the top of the SR curve here. But when we've analysed this, it appears that these compounds are kind of producing an intermediate. It is less than the extent of offness. That's a new word. That I know new words are trendy in America at the moment. That it's not as far off as the SR seems to be turning the receptor. And that actually fitted with some of the modelling data that was coming out at the time that was suggesting that org and PSN were kind of producing an intermediate off state of the receptor. And so this sort of fitted with that as well. So does org do anything on its own? Is there any of this allosteric agonism that we could see? So at very high concentrations here, this is 30 micromolar now, we do see inverse agonism in response to org. Now, there we go. If you look at that compared to, here's in the presence of the CP at 30 micromolar, where it's pretty much instantaneous, they're reaching the same point, but it's happening much, much faster if the orthosteric ligand is actually there. So we tend to think of allos modulating the activity or the binding of the orthosteric ligand. We think here the orthosteric ligand is actually modulating the binding of the allosteric site. And it makes absolute sense that, you know, these things could work in both directions, because we're clearly seeing a difference here when the orthosteric ligand is present. And so when we analysed that and wondered, is it quicker because it's more potent? Because remember, we saw that as you went to higher concentrations, it happens faster. So these are a little confusing to look at because they don't start from the same place. So org alone, you're starting at the forskolin, and then you're seeing only at very, very high concentrations do we see this inverse agonism. In the presence of CP, we've inhibited the cyclic AMP, and then as we add increasing amounts of the org, you can see it is a lot more potent in the presence of the orthosteric ligand than it is on its own. This is, of course, the exact opposite of what you see with a competitive ligand. So when we look at CP alone versus, SR alone versus the SR in the presence of CP, you can see that on its own, it is much more potent than it is when you have an orthosteric ligand agonist present. And of course it is, right? They're competing for the same binding site. It has to outcompete the agonist before it can have a response. So this is your classical pharmacology, and this is kind of cool, I think, because it shows you that this is genuinely allosteric. It's acting in the opposite way, and we're looking at conformationally linked sites here. So then we were interested in this idea that the org is turning the receptor off in some way. Is it desensitizing the receptors? What is it doing to them? And we teamed up with Mark Connor at Macquarie University, who had a PhD student, Will Redmond, who's done this work with us. Now they were looking at ATT20 cells in which you can see the potassium channel response, those inwardly rectifying potassium channels that are activated in response to the cannabinoid activation of the G protein. And they're doing a fluorescent assay. You can see very rapid activation of this potassium channel here, and then with the orthosteric, with CP still, you see this happens rapidly, and then the receptor very, very slowly actually desensitizes in these cells, very slowly returns back. When they added org into this, this is at 10 micromolar org, you can see that maximum inhibition still occurred, and then the receptor got turned off much more rapidly. So it really sort of fit with what we were seeing with the cyclic AMP assay, that there is this delay. The maximum response wasn't changed, but the rate at which the receptor desensitized appeared to be much greater, much faster. So desensitization is one step on a pathway, right? And this is the sort of canonical signaling pathway that we're expecting these receptors to follow. That's the agonist is activating the receptor. That leads to activation of the G protein. The receptor is then phosphorylated by the GRKs. That leads to recruitment of arrestin, which is the first step in pulling a load of different adapter proteins together and internalizing the receptor. And so we were seeing decreased signaling here, but we seem to be seeing, rapid desensitization. So we thought, well, that should result in really rapid internalization of the receptor if this is following a classical pathway. So we had a look at what was happening in terms of internalization. So CP55940 produces rapid and potent internalization of the receptors, such that we see pretty much 100% internalization. This is an hour drug treatment, but as David Finlay will present a bit tomorrow, within about 20 minutes, the receptors are fully internalized with CP. So then we looked at what the allos did to this. So we've internalized with the CP, and then looked at different concentrations of the allosteric modulator. And you can see rather than enhancing the internalization as we'd initially expected, they actually appear to block the internalization of the receptor. And when we looked at the rate of internalization, you can see the CP rate here, very rapid and quick, and it looks like they're just sort of preventing the extent of internalization that we would normally see rather than particularly slowing it. It's not just slowed down, we do seem to have plateaued by the end of this. So we've got enhanced desensitization, less internalization. It's not all fitting together at all nicely. And around this time, Romano Silvestri with Vincenzo Di Marzo had reported a whole series of analogues of the Organon compound. And so we thought, well, can we tease apart some of this mechanism by looking at these other compounds? And we initially looked at them in our cyclic AMP assay and looked at that delay. How much of a delay was there before they turned the receptor off? And what we saw was with some substitutions, we were seeing very, very long delays. All of these compounds were producing antagonism, but some of them, you really had to be patient to wait before you would see it. But a series of them appeared to be producing absolutely immediate blockade of the receptor. So we thought that was really interesting, and we submitted it. And again, the referee saved us from going wrong with this by saying, well, we want to see the potassium channel assay and see what's happening in that. And so we called Mark, and he very quickly did these assays for us. And what we did was we took a series of the compounds that appeared to be producing immediate responses in our cyclic AMP assay, and we looked at them in his GERK channel assay. And we don't need to go through them all in detail. I think this was compound 1, which was the Org compound, but these were some of the others that were sitting at this end. But with all of them, we saw absolutely no change in the maximum response. But they all produce this rapid desensitization of the receptor. And you can see them shown here with the peak hyperpolarization not changing for any of these compounds. But with SR, when you're antagonizing the receptor, the peak is clearly decreasing at each of these concentrations. So these are a really different mechanism of action to a classical antagonist, I think. And what it's sort of suggesting to us is this assay is way more sensitive because this initial peak is so rapid. It's more sensitive than the cyclic AMP assay is to these sort of subtle changes in the peak response. So as a sort of interim summary of where we've got to here, these are clearly not acting as just non-competitive antagonists. They're not just turning the receptor off. They appear to be doing it in a way in which the early signaling is unaltered, but then the receptor gets turned off. And we see a reduction in constitutive activity. So there is some inverse agonism. And when we've looked at this in a series of compounds, we can produce responses that, at least in the cyclic AMP assay, are very similar to what you would see with a classical inverse agonism, but the potassium channel assay is telling us that no, this is a very different mechanism of action. And so what we would really like to know is what's the impact of that? So 5-minute cyclic AMP signaling, how different is that in terms of a behavioral response than 20-minute cyclic AMP signaling? And so far, I think no one's really been able to tell us because these compounds are not translating particularly well in vivo. So Aaron Lichman's group did a lot of work with the Org compound, and the only behavioral response they saw was not receptor mediated. And so there could be lots of different reasons for that. It could be that they're just not potent enough. There's still, we're only seeing these effects at micromolar concentrations. And so it may be that just in vivo you're just not getting there. It could be pharmacokinetic. We don't know, but we think that it would be, if we could see this it would be a really cool way of actually teasing apart what the early signaling is mediating versus what later signaling events are mediating. And in terms of mechanism right now, we just think that they're turning the receptor off. We don't think that they're doing anything particularly intricate in terms of the desensitization pathway. We just think they're pushing it into an off conformation. So that's not driving, that fits with it, not sort of driving the internalization, because it's just turned off rather than being downstream of activation like classical desensitization would be. So are these compounds biased agonists? This has been the big question, I think, around these. And around this same time as we were doing that initial work that was published in BJP, Deborah Kendall's group was doing this work in looking at these compounds. And in a series of papers, what they suggested was that ORG alone could drive G-protein independent phosphorylation of ERK pathways. And then they went on to suggest that this was actually being mediated by beta-arrestin 1, and that ORG was a beta-arrestin 1 biased ligand. And for those of you not sort of in the receptor signaling area, this idea of bias is a very exciting one in pharmacology right now, that we might be able to drive subsets of pathways rather than all possible pathways by just driving a particular conformation of the receptor. And it's the same idea as really as the allosteric modulator of if we can do less with a compound, maybe we will get less adverse effects. We can find the right pathways to activate. And so just showing the sort of we thought this was really nicely done work. They do lots of great controls for it. Here's the CP response at 5 minutes, and the ORG was producing a response that was at least double the size of the CP response, and was actually, CP was in a way inhibiting that. And they looked at it in the beta-arrestin 1 knockdown, and they showed that the response to CP is, as expected, G-protein mediated, but the response to ORG was diminished in the beta-arrestin 1 knockdown cells. And so suggesting that ORG was driving this through an arrestin pathway. But what do we see in our hCB1 cells? We see a response to CP, that's phospho-ERK activation here now. This is time dependent, it's a very transient response. But you see a really robust peak signal. This is at 20 nanomolar. Peaks that are sort of between 3 and 5 minutes. That actually changes day by day. But around 3 to 5 minutes. We can block that with ORG, which we would actually expect. It blocks GTP gamma S binding. It blocks cyclic AMP signaling. And well, you know, it also blocked this response. And on its own, which is where they were seeing this dramatic phospho-ERK stimulation, we see absolutely no stimulation of phospho-ERK in response to ORG alone. And we've done this at a very wide range of concentrations, and in no circumstances is it driving it. So I get a bit obsessive about lack of reproducibility between labs. I hate it. And one of the reasons is I really like HEC cells. I like them because we can dissect signaling out. And we can ask questions in a HEC cell that we can't ask in cells with really little receptor in them or whole animals. You know, you never see a 5-minute delay in signaling unless you're using some sort of sensitive model. But the only way a HEC cell can be a good model is if my data in a HEC cell looks like your data in a HEC cell, and we can agree on these things, and we can agree on basic mechanism of the drugs, and then we can take that forward. And so somebody else's HEC cell doing something that mine doesn't really drives me a bit nuts. And so Deborah had done all of that work in hCB1 cells, so we should have been seeing it, and it should have been a neat launching point for other studies we were going to do, but we could not see it. And so I wanted to know, why can't we see it? And if we cut to the punchline, I can't tell you why we can't see it, but I'm going to show you all the efforts we've made to see it. So we looked and chose CB1s. We looked in every cell we have in the lab, basically, that has CB1, that we put in CB1 or has native CB1. So we've looked in CHO CB1 cells, we've looked in our different HEC backgrounds, the HEC flippins, the HEC FTs, the ATT20s, neuro2As that are endogenous mouse CB1, and even at the time we were doing some work with some primary human glioblastoma cells. They have endogenous CB1. They respond, here's two of those human cell lines. They respond to CP and generate a phospho-ERK response. This can be blocked with SR or with ORG. In both cases it was blocked by ORG, and in both cases we didn't see, in both of these cell lines, we didn't see any effect of ORG alone. So I know this is similar to what Tom's reported recently as well, and we had similar questions to clearly what he was having at the time, which was, is it because we're doing the assay by alpha screen? So alpha screen is great, it's horribly expensive, but it's a really quick way and very sensitive to measuring phospho-ERK. But you lyse the cells in a very gentle lysis buffer. So you're only seeing cytoplasmic ERK. And so we thought, well, could this beta-arrestin signal be nuclear? And therefore we're missing it entirely in our assays. So we have a high content immunocytochemistry assay that we can use instead, where we can actually differentiate cytoplasmic from nuclear staining, and when we analyze them separately, much like Tom did, we still don't see it. We've tried multiple batches of ORG. We've had Romano Silvestri's ORG as well that we were using for his compounds. None of them are driving this. So it's not just that we had a bad batch or something as well. So we tested lots of batches and different suppliers as well. So what we didn't have when we were first trying to solve this, though, was a nice arrestin recruitment assay. But we've recently got one of these. And so this is Jonathan Jarvich's assay. And this is quite different to the other arrestin assays being used around the place, because the receptor is not tagged in any way. So you still just have your receptor in the cell membrane. And what is tagged is the cell membrane is tagged with a citrine, and the beta-arrestin is tagged with luciferase. And so you transfect all of these things in. And we feel this has a number of advantages over the other assays. Firstly, the receptor is not tagged. And with CB1, we think that's really important because, well, for starters, you're putting a giant fluorescent tag normally on the C-terminal, which is precisely the part that you think is mediating this signaling. So it's always sat a little bit uncomfortably. But we also get really poor expression of fluorescent CB1, the bulk of it being intracellular. And one of the advantages of this assay is you're only looking at interactions that are occurring at the cell membrane rather than potentially more the fluorescently tagged CB1, your cytoplasm is just jam-packed full of fluorescently tagged CB1. So that's an advantage. We can do no receptor controls as well and see if any of the signal is non-specific, and that's really important with Brett because just conformational changes will result in changes in Brett signal. So it's really important to have these controls available to you. And then what we feel is the real advantage is it actually gives you the ability to compare between receptors. So generally with a Brett assay, you can't compare between the size of the signal that you see because it's about the conformation of the receptors. And you can get people have shown that you can get an increase in Brett or a decrease in Brett just by switching the tags to the opposite proteins. Right? So the size of the signal is not thought to be relevant. But in this assay, so long as you're controlling for the expression of your Brett constructs, the size of the assay should reflect how much the arrestin is translocating to the cell membrane. So we think it's got some advantages over some of the other assays. There it is. So it's translocated. And now we see the ability to resonant energy transfer to the fluorescent molecule and emit a signal there. So starting with the vasopressin receptor, which we know is an excellent recruiter of arrestin. This is with bovine beta-arrestin 2. So you can see again time. We're adding the drugs at time 0 here, and it's a change in the Brett ratio relative to that point. And you can see that with this V2R agonist, We see very potent, rapid increase in arrestin translocation. At higher concentrations, it gets more rapid, but we're kind of reaching the same maximum point there. And they're relatively stable across 20 minutes of the assay. Traditionally, arrestin recruitment assays are done with bovine beta-arrestins. We've never known why that was. So we've made human beta-arrestins in all of this, and they behave identically to what we're seeing with the bovine. So then we came to look at CB1 and arrestin recruitment, and just starting with the beta-arrestin 2. And this is with WIN, which I'll talk more about this tomorrow, is one of our more efficacious ligands in recruiting beta-arrestin 2. And you can see that we do see an increase in the arrestin 2 translocation relative to the vehicle response. And when we add the org into this, with org alone, we see no movement of the arrestin at all. And when we add it with the agonist, we're definitely blocking that WIN response, and we may even be blocking a constitutive activity here. But this is quite noisy data, right? And this is why this is quite noisy data, and I will talk more about this in my talk tomorrow, is because if you look at it in scale, the size of the response that we are seeing in response to a cannabinoid is tiny compared to what we are seeing in response to other receptors. It is really small. So we are sort of pushing the limits of our signal-to-noise detection here. But nonetheless, we can do an area under the curve analysis of this, and area under the curve sort of, will magnify sort of small differences between curves, really. Oh, no, I'm not going there. What about beta-arrestin 1? So the other lab was suggesting that org was going to be driving a beta-1 arrestin recruitment to the receptor. So here is our beta-arrestin 1 signaling with CB1. Tomorrow I will prove to you that this assay is actually working with some other ligands that are driving this pathway. So today, in the interest of time, you kind of have to take my word for it. Here's our no receptor control, and you can see it's tidier. And so we did wonder, is there something here, and it's just so small that we can barely detect it? If you look at this on this one day, you can see the no receptor control versus when we have CB1. There is maybe a difference between those. Here's just 3 repeats so far, looking at with CB1 versus no CB1. We're going to have to do a lot of ends, I think, if we're going to see anything statistically significant within this. So if it is recruiting, it is very, very subtle in terms of this, this recruitment. So then we thought, well, it was still, we're still thinking, and this is actually before Tom's paper came out, that we were still thinking, we should publish this though, right? We should tell people that we can't see this, because lots of people are building org-like compounds thinking that they're going to be biased, and yet in our hands, they're not biased. So we should tell people, and I was trying to think, for me, publishing negative data is really uncomfortable. Publishing you saw it and we don't is really uncomfortable at times. So we thought if we could explain the difference, then it would be okay. And we could put it out there and say, we don't see it, but here's why we don't see it. And so our final question was, in terms of trying to understand this, could the cells just have really limited beta-arrestin 1? And we've got lots of receptor there, and maybe it's just not enough. And we've done this PCR in these cells now, and this is 35 cycles, so you really should be seeing it if it's there. And we looked first in our standard cell line that we use, and there's a little faint band there. And we sort of panicked a bit because, like many people's, these hex cells have been in my lab for a very long time now. We don't really remember where they came from because we kind of got them at a time when record keeping was not as paranoid as it is now. But so we bought new hex cells from ATCC. These are sort of brand new. We know where they've come from, and they're definitely, hex cells. And it's not really there either. There's a very, very faint band. So we thought, well, okay, we've got the answer. the cells are limited in beta-arrestin 1. We're going to put in more arrestin 1 and wham, we're going to start seeing the signaling. Nope. We made a stable transfected beta-arrestin 1 cell line. And then we've looked at the signaling of the phospho-ERK, and we've done it across a longer time point because there's been suggestions that beta-arrestin signaling might be more delayed here. And you can see our CP response. It does seem to be slightly blunted in our beta-arrestin 1 cells. But the org did nothing again. If anything, there may be a slight inhibition in these. The org has done nothing. We know HEC cells are supposed to be limited in GRK2, and that is important for arrestin recruitment of some GPCRs. So we've also added additional GRK2 into this, but that also does not appear to be the answer, and I'll show you some more of that data tomorrow. So there is nothing we can do to these cells that are going to get them to drive this particular pathway, we have decided. And I would be willing to accept that maybe it's a limitation of this HEC cell because maybe we're lacking some other adapter protein, but I just don't believe that every cell in our lab is lacking the critical machinery to drive this pathway. So in conclusion here, we really strongly believe that these compounds inhibit G protein signaling, and they do this in this very cool time-dependent manner where they appear to increase the rate of the desensitization of the receptor, but I'm using desensitization quite loosely there. They turn the receptor off. They're clearly not doing this with classical desensitization pathways because they're inhibiting the recruitment of beta-arrestin 2, and that is part of your classical pathway. So they are preventing internalization of the receptor in a way that is clearly not being mediated by arrestin. So what it would appear, and again, touching back again on some of this tomorrow, is that this internalization pathway is not being mediated through, is not arrestin mediated. And to date, we can find no evidence for non-G protein mediated signaling. If you guys have the answer to that, we would love to have it, and then I can let this go. But you know, for now, we see no evidence of bias signaling. Everything we see is completely consistent with the org turning off the receptor. And so that is it. I just want to particularly acknowledge Erin Corsten, who's left my group now but did much of the early work on these org compounds. This is my group, and this is Tash Grimsey, many of you know, who has a sub-team within my team, so half of these people are hers. So just to particularly acknowledge David and Mikkel and Krista, who have all contributed data to this talk today. And thank you very much for your attention, and I'm happy to take questions. Just got one.
My name's Dow Hurd. I was wondering, have you thought about looking at whether you have GERK6 concentrations in your cells?
Yeah.
Okay.
Yep. So I'll show you tomorrow. We have tested GERK6 in this as well. We've done 2, 6, and 5, I think, so far, and it's not the answer yet.
Very nice. As always, seeing things in real time unveils complex interactions, and so you've really shown me something interesting. Have you thought about alpha-beta effects? And so alpha will increase the rate of onset of your agonist, and that will increase the onset of your org, and then as it binds, it cuts off signaling. So have you, when you showed that range of delays, do you have a set of relative alpha beta values for those compounds? Because you could imagine that that's doing something with the real time, changing rates of onset, signaling, and so forth.
So no is the very short answer there. We've characterized the compounds within the binding assays where you see this enhancement of the binding, and the ones that we went forward with had similar is it potency in that case, a potency to org in that, and yet you were seeing the delays, and likewise with the PSN. So I'm not answering your question.
I mean, values of alpha.
Yeah.
Potency is
Separate, yeah.
Fine, but values of alpha beta, because, well.
Yeah. So no.
It's interesting to see if that range in delays
correlated. Yeah, we'll have a look. Have a look when I get home. Yes.
Well, I would just comment, this sequential process that you see, speaks confirmations, opening for the allosteric modulator and then that desensitizing it. So I really like that line of thinking. And I was also interested in, I don't know what this means, but what the height of the plateau, how that related as well, as to how much CP55940 might have to bind to open up this allosteric. Was there something that it was a stepwise relationship there? So I'd really, the question about the alpha beta and what does that look like, and then what is driving the height of that plateau would be something else. It's very fascinating data.
I think, the height of the plateau we have looked at more, and we've been looking at some compounds from Chris Cunningham that are org related as well, in which we see a very similar length of delay, but we're not going above forskolin. So they're turning the receptor off, but they don't seem to be driving it fully to inverse agonism. So I think there's some interesting stuff there.
It's a question I don't know, obviously, but it was intriguing to think that while it was tracking along the CT, was that part of what was driving the plateau. So
Right.
I'd like to see that.
The comparison between the delays and which I think all that data would be within that MedChem paper, but gosh, there were a lot of compounds there. So we should look at that again.
You seem reluctantly almost to publish your data. Are you aware that anybody has unpublished data in some other GPCR that saw what you saw?
Well, actually, because Tom here has published that he couldn't see this either, and so now we're sort of thinking, well, do we need someone else to say they can't see it, or what do we do? We're still looking for that explanation, really. But I'm sure there is a lot of. We know that there are a lot of great papers out there that people can't reproduce, and I think it's important that you tell people that you can't.
Yeah,
it
I
saves
agree.
people wasting time. So we were, we just sort of really wanted to explain it rather than just say we can't see it. I think Patty's got a question here.
I know the reviewers got you to do those ATT20 cells
Yeah.
in the potassium channel. Do you know how much beta-arrestin there is in ATT20s? Beta-arrestin 1 and 2?
Yeah. Have we checked in our hands? No, we don't.
I think Ken Mackey published a paper.
Yes, but
where
so
he
the
gave the
Atwood
rate.
paper says that Hex have 1 and 2.
They have both.
Yeah, which they don't. I'll show you tomorrow.
Okay.
They don't.
Oh, Lord. Well, according to Ken
So you all have
Mackey,
to come tomorrow, right?
I think beta-arrestin 1 is really low in ATT20s.
But they did supposed to have 2, and we do think 2 is what is more important in the internalization pathways.
Yeah.
Whereas 1, I'll show you tomorrow.
Okay.
I'm going to steal all my stuff from tomorrow.
Hey, Michelle, so that was great. Is this on? Can you hear me?
Yeah.
So it's nice to see that I'm not crazy. But one thing, going to what Terry was asking about looking at the alpha for those compounds, so did you look at org against any other agonists? Because it does seem that CP and org do have a lot of a stronger binding cooperativity maybe than some of the other agonists.
Yeah.
And maybe if you saw differences based upon what your agonist
So we
was.
looked at it with with 2AG and with anandamide, and with THC, as well. And there were subtle differences. The thing with org is it's quite low potency. And so you sort of, because we're not reaching maximum effect generally at 10 micromolar, and we really can't go higher than that without it falling out of solution. So we could get to about 30, but that's as far as we trust it to go. And so, we can see subtle differences in that between the different agonists when we match them for efficacy. So if we do it at EC50s of all of the agonists and then look at the org responsiveness, we do see subtle differences between them, but nothing particularly dramatic, nothing that would say it's going to turn this off but not that. They were maybe half a log. Yeah. I think we might be done.
I am President and Chief Executive Officer. I'm one of the social scientists. Your presentation.
Thank you.